interfaces are required for selective storing.
Right.
So
Right.
what I don't understand is the dynamics, why you consider that equally fundamental. So in particular cycles, I know in the particular experiments that we talked about, there are cycles. Washington, right? But are you confident that this is a fundamental abstract ingredient for originating life? Or is it just the way that you see that it happens in the experiment?
Cycles are the absolute best way to select from a system, because you have a system as you heat, as you cool, wet and dry. Think about the polymeric chain reaction if you're familiar with PCR. But over and over again, when you do cycles of one sort and another, and there are many different kinds of cycles on the early Earth, you have tremendous selection mechanism. You winnow things out at high temperature, you winnow things out at low temperature, you winnow things out when they're wet, you winnow things out when they're dry. And as you cycle through a system of chemicals, what's left over is the things that are robust enough to survive that. And that really is, so I think cycles really are fundamental to the problem. Without cycles, I don't see that you have sufficient selection mechanisms to really drive things along. So, and I think that if you talk to evolutionary biologists, there are other kinds of cycles in perhaps a much longer time, but cycles really do drive things. Cycles of change. Yes?
I do have a question, but I also wanted to add that cycles also provide energy into the system. So as the wave comes up the beach, you have the energy moving the sand grains and the ripples.
Yes.
I agree with that. I was also wondering about when you were talking about how you're trying to get in the laboratory the reverse citric acid cycle. So you're trying to get that going in a laboratory setting and show that it can emerge on its own. And to do that, are you sort of taking a molecular evolution approach, or does that not really apply to something as complex as this kind of cycle? And if not, then how do you go about doing something like that?
In these experiments, which are spearheaded by George Cody, what we're trying to do is look at each step in the citric acid cycle. Say if we start with aconitic acid, can we go to the next step? Can we go around? When we get to citric acid, can we see that it splits into 2+4 oxaloacetic plus acetic acid? Can we see each of those steps occurring? I'm not trying to get the whole cycle to run as a continuous, but actually just step by step looking at what each of the components of that cycle does in a chemical sense. So that's a very preliminary state. I mean, we're nowhere close to actually getting a bunch of chemicals in an environment and running a TCA cycle. No enzymes, except sometimes we have minerals. And minerals are quite amazing. The iron and nickel sulfides, cobalt sulfide, for example, can really promote some of these reactions. So we only allow us geochemically plausible minerals, no enzymes. Let's see, let's take the question here. Yes, sir.
I have a question that harks back to what you said, your definition of intelligent design at the beginning. And personally, I don't see a conflict between what you've defined intelligent design and evolution, because an intelligent source could have planted a primitive cell on the Earth, and then evolution takes over and then explains how you get from the cell to the human. And I think this is, an ongoing debate in this country with religious fundamentalists and so on. This thing gets sort of mixed up and confused in the issue, but I think the two things can be separate. I mean, if you see what I'm getting at, maybe it's primarily semantic, but
for the origin, if you say the first cell was a miraculous or a planted thing or designed or something of that sort, and there's really nothing a scientist can say about that. If that's the way it happened, and no one in this room can disprove that hypothesis. There's absolutely nothing we can do to prove this. But what we can show, I'm hoping, is that there's a chemical, physical process where you can go to the first cell. And if that's true, then I don't see the need for the other. But it's true. I mean, science really is impotent when it comes to disproving any of these other hypotheses. That's not what science does. What science does is show what's plausible using the laws of chemistry and physics. And that's really the only thing I can comment.
I agree with you, and I certainly 100% behind what the technical part of your talk, but I'm harking back. It's not directly related to your theme, but this debate, because you can still have intelligent design and Darwinian evolution. Is what I'm trying to
Yes.
say. Okay.
Let's see. Here, please.
What do you make of panspermia?
Panspermia is a fascinating concept that life on Earth was seeded from someplace else. Of course, in terms of origin of life research, it just deflects or defers that origin event to some other place and time, perhaps a place with a different chemistry. The most active aspect of panspermia has to do with Mars, because Mars was a warm, wet planet before Earth, potentially. If origin of life occurs quickly, that is within a few million years or tens of millions of years, one can imagine life originating on Mars, microbial life, and then a large impact on Mars could have thrown meteorites which contain relatively undisturbed life forms. They could have traveled in the flux of meteorites that goes from Mars to Earth, and we know such things are here. And Earth could have been seeded with microbial life from Mars before life really got going here. That is actually a very plausible hypothesis that a lot of scientists are thinking about. It's one of the rationales NASA uses to say we got to go to Mars because we could all be Martians, in a sense. More remote kinds of panspermia, or the directed panspermia where some intelligent civilization sprinkles cells on promising planets and then just lets them take off. Again, there's no way that we can prove or disprove that. I don't see a particular need for it. It's fascinating, and it's just one of those hypotheses that's out there. It's kind of an intelligent design, but maybe one that's more plausibly explained. And I suppose I can imagine a human civilization far in the future where we have space travel, trying an experiment like that. You find a nice Earth-like planet where there's no life, and you sprinkle something and just sit back and see what happens. But you know, planetary protection officers at NASA would probably be a little upset at that. Let's see, you had your hand up for a while.
When we do, assuming that we do figure out the actual origin of life and everything that happened, how do you think that will affect our search for life on other planets or bodies?
They may be sort of decoupled. I think if we show that there is a very deterministic process, that is, the origin of life is a cosmic imperative, then it will be more and more urgent to have perhaps a human mission to Mars, because the kinds of search that we'll want to do involve drilling and so forth would certainly, I think, be very exciting, and it's hard to do that with a rover. But you know, there's laboratory-based experiments on Earth, and there's now the other possibility is if we go to Mars and find that there's life all over the place and it's different from Earth life, then that gives us a second biochemistry and may give us some more guiding principles. Because one of the biggest problems with the origin of life is we really don't know what life is in terms of rigorous definition. Would you agree, Lynn? I mean, I think we don't have an absolute ironclad definition because we don't know the full range of things that might be living or not. But
we don't have anything absolute.
Nothing absolute. Yeah. So if you ask 10 biologists to define life, you'd probably get 10 slightly different answers. Yes, please.
Would you say our understanding of how we get from prebiotic chemistry to self-replicating systems is far enough along that we're just waiting for someone to just do the right experiment for it to come together, or are we missing something fundamental?
I hope we're far enough along, but if we're missing something fundamental, we're not aware of it. Is the trouble. So I think a lot of people understand the principles, and in fact, the real stumbling block for a lot of people is just how do you synthesize RNA in a prebiotic environment? And that seems like a fairly simple chemical. And there's new ideas that are being approached right now to try to do in what's called a systems chemistry approach, as opposed to the old building block approach where you take a sugar and you try to stick a phosphate on it, and you try to stick a base on it, you know, and 1, 2, 3, and that doesn't seem to work very well. But there may be other ways, or there may be other RNA-like molecules, which a lot of people have proposed, and I think that's still being explored very intensively by the community. If that works, then people will say the problem has been solved. A lot of details to fill in, though.